All right, so we're going to begin our discussion of the various organelles by talking about the nucleus and the ribosomes. The nucleus is going to be the command center of the cell. It's going to be found usually near the center of the cell, and it's going to be separated from the cytoplasm by a nuclear envelope. This nuclear envelope contains a double layer of membrane. So if you remember when we talked about the plasma membrane and how the plasma membrane is a lipid bilayer, so it's made out of, well, this is a double bilayer. So there's actually like a 4-phospholipid membrane thickness found in this nuclear envelope. It's going to consist of a double layer of membrane. It's going to have nuclear pores that permit the exchange of nucleoplasm and cytoplasm. These nuclear pores is also where RNA is going to leave the nucleus. Once inside the nucleus, DNA does not leave that nucleus. It's safe in here. That chromatin, that information is found bound tight, nice and tight. It's safe inside the nucleus. It's not going to leave the nucleus. However, we do need that information to leave. So although the DNA does not leave, our hard copy does not leave the nucleus, we do need that information to leave the nucleus in order to turn it into proteins. We're going to do this by copying down that information from DNA into a message known as RNA, and that RNA is what's going to actually leave the nucleus through those nuclear pores. Inside the nucleus, we're going to find chromatin. Chromatin is going to be contained in a semi-fluid known as the nucleoplasm. The nucleoplasm is kind of like the cytoplasm. It's the jelly-like substance that's found inside the nucleus. The chromatin is going to contain nucleic acids and proteins. This is going to condense down to form chromosomes during our cell division. Chromosomes are going to be the carriers for our genetic information. Now, there is a region inside of the nucleus, a much darker region known as the nucleolus. The nucleolus is going to be composed of ribosomal RNA. The ribosomal RNA is going to be responsible for making the ribosomes. So ribosomes are our protein factories. The nucleolus is our ribosome factory. So here's our nucleus. Like I said before, it is physically separated from the cytoplasm by a double membrane known as the nuclear envelope. It is still able to communicate with the cytoplasm through those nuclear pores. And like I said, those nuclear pores are large enough to permit the passage of ribosomal subunits and messenger RNA out of the nucleus, as well as the passage of proteins from the cytoplasm into the nucleus. It kind of acts as the gatekeeper and regulates the materials that go into and out of the nucleus. Next up, we have the chromatin. Chromatin is going to be found in the semi-fluid known as nucleoplasm. So like I said before, that chromatin is going to contain the nucleic acids and proteins. It's going to condense down to form chromosomes during our cellular division. Finally, our nucleolus. Like I said, it's the darker region of the nucleus, going to be composed of rRNA. This is going to be responsible for producing subunits of ribosomes. Ribosomes, as we kind of already mentioned, play a very important role in our passing of our genetic information on into its next form, proteins. So these are going to be ultimately composed of RNA. They're often called the protein factories of the cell. They're going to be composed of a large subunit and a small subunit, and they're going to be made inside of the nucleolus. Now that's where they are made. However, they primarily are located outside of the outside of the nucleus. They're going to be found, for the most part, on the endoplasmic reticulum, or free-floating inside of the cytoplasm. The number of ribosomes is going to vary. Number of ribosomes in the cell is going to vary depending on its function. For example, the pancreatic cells have a lot more ribosomes because they are responsible for producing protein secretions like insulin. If we have a lot of product to produce, we're going to need a lot of factories up and running, working night and day. And so our pancreatic cells are going to have a lot more ribosomes that are going to be constantly working to produce insulin. So like I said, they are going to be found either on the endoplasmic reticulum or free-floating in the cytoplasm. Now, like I said, this is going to be the site of protein synthesis in the cell, and we will have an entire chapter devoted to this, but I do want to just touch on it now so that way when we do talk about transcription and translation, you'll have at least seen it before. So these protein factories, this is where these protein factories, all of this process of producing proteins is going to start off inside the nucleus. Then inside the nucleus, the genetic information is going to be first copied down as mRNA. Like I said before, you can kind of think of DNA as your textbook. It's a hard copy. We don't want to change a word of it. We don't want to risk damaging it. So if you are going to utilize it, the first thing you want to do is copy down and take notes. So we're going to copy down genetic information just on the areas that we want to discuss. If we want to make a tiny little protein, or if we want to do the chapter 1 homework assignment, we are not going to copy down the entire freaking textbook. That is unnecessary. We don't need the whole textbook. We don't need a copy of the entire textbook. We only need a usable working copy of the most important information that we need to obtain. So we're going to do this by copying down that information in shorthand notes, otherwise known as mRNA. That mRNA is not our hard copy. So we can risk it leaving the nucleus. It really only exists for as long as we need to transmit that information from the nucleus or from the DNA into proteins. So we really only need to exist and travel from the nucleus to a ribosome. So once we have copied down our information in mRNA, we are going to leave the nucleus through the nuclear pores in the nuclear envelope and go into the cytoplasm, where we're going to meet up with the ribosome. Once we're at the ribosome, the site of protein synthesis, those ribosomes are going to read the genetic instructions of that mRNA, and then they're going to be responsible for producing a polypeptide strand. If you remember, when we talked about a polypeptide strand, that is only one level of the structure of protein. So see if you can find out, without looking at your notes, what level of structure is the polypeptide. Is that the primary level, secondary level, tertiary level, or quaternary level of organization of a protein? That's it for our discussion on nucleus and ribosomes. Please let me know if you have any questions.